Hello, my name is Daniela Anderson, and as part of Dr. Alexa Venema's lab, I will be presenting my investigation on the role of oxytocin receptors in the sex-specific regulation of social behavior in juvenile rats. The oxytocin receptor system is a crucial component for the regulation of a broad range of social behaviors in both males and females across a variety of species. In juvenile rats, oxytocin receptors are expressed throughout multiple brain areas involved in social behaviors. One such brain area is the ventromedial hypothalamus, or VMH, which is involved in the regulation of an array of social behaviors, including social investigation. When we look at oxytocin receptor binding densities of rats in the VMH, adult males show a higher binding density than adult females do, indicating higher expression of oxytocin receptors in adult males compared to adult females. However, when comparing oxytocin receptor binding densities between male and female juvenile rats, we see no sex difference. With this information in mind, we sought to determine whether cells that express oxytocin receptors in the VMH are recruited in a sex-specific manner following an exposure to a social stimulus. Since we expect no sex difference in the amount of oxytocin receptor expression in juvenile rats, we hypothesize there will also be no sex-specific recruitment of cells expressing oxytocin receptors in the VMH following exposure to a social stimulus. In order to test our hypothesis, we used a group of 20 male juvenile Wistar rats and 20 female juvenile Wistar rats. 10 rats from each sex group were exposed to a social stimulus, while the other 10 rats were left alone. The social stimulus consisted of placing the test subject in their home cage with a 3-week-old sex-matched stimulus rat for 4 minutes. 30 minutes following social exposure, when levels of CFOS mRNA peak following neuronal activation, brains were extracted. The fresh frozen brains of all 40 subjects were blocked off at the VMH and cut into 16-micron sections using a cryostat and mounted on slides. The ACD bioRNAscope system was then used to perform fluorescent in situ hybridization to target CFOS and oxytocin receptor mRNA. Images of the slides pertaining to bregma distances -2.40 and -2.52 were used to count the number of cells that express CFOS mRNA, oxytocin receptor mRNA, as well as cells that express mRNA for both CFOS and oxytocin receptors. Cells that express CFOS indicated activation of the cell. Oxytocin receptor positive cells showed expression patterns of the oxytocin receptor system, and the presence of double positive cells indicated the specific recruitment of cells within the oxytocin receptor system of the VMH. In Figure 1, as was previously expected, we can see no sex difference in the number of cells that expressed oxytocin receptor mRNA within the VMH of juvenile rats. In Figure 2, we can see females showed a greater number of CFOS positive cells, indicating an increased level of VMH activation compared to the males, regardless of social exposure. In Figure 3, we see a trend for a significant sex difference in the effect of the exposure to a social stimulus, where females expressed more CFOS and oxytocin receptor double positive cells than males in the VMH when animals were exposed to a social stimulus. This could indicate that female juvenile rats recruited more oxytocin receptor expressing cells once they were exposed to the social stimulus than male juveniles did following the same social exposure. In our study, we established a clear trend relating to the role of oxytocin receptors in the sex-specific regulation of social behaviors in juvenile rats within the VMH. Our findings show that even though there is no sex difference in oxytocin receptor expression, females exposed to a social stimulus are more likely to recruit oxytocin receptor expressing cells than males exposed to the same social stimulus. Our future studies will focus on determining the role of oxytocin receptors in the sex-specific regulation of social behavior in both adult and juvenile rats within the bed nucleus stria terminalis, the medial amygdala, and the VMH. A correlational analysis between these highly important brain regions involved in the regulation of social behavior will reveal whether activation of oxytocin receptor populations within these areas show coordinated activity following exposure to a social stimulus.